Okay, so the question on page 164 is talking about fruit flies, Drosophila. Now we love Drosophila because they reproduce really quickly and they've got diploid chromosomes, and usually the genes that we look at are expressed with single allele or maybe two. So they're very easy to study when we're looking at genetic tropes. We're talking about a pure breeding variety with red eyes and tiny wings. So what I'm going to do is I'm going to add in some red eyes on my little Drosophila and some vestigial wings. And don't laugh at my drawing. With a pure breeding variety with pink eyes and normal wings. So we didn't get laughed at. All the F1 flies, and these two were crossed. So these two parents were crossed together, and they produced a range of offspring. All the F1 flies had red eyes and they all had normal wings. So my F1 generation has taken red eyes and normal wings. Now if we think back to that question we were looking at when we're looking at our pure breeding black dog, a black haired dog and red haired dog, we've got the same kind of a situation here. We've got a dominant allele and a recessive allele, and when we mix the pure breeding types, the dominant allele is shown. What happens next though is when we interbreed or intercross two of these alleles, then we get a mix in our F2 generation, and they produced different results. So we had red eyes with vestigial wings, so we have some red-eyed flies with tiny wings. We had some red eyes with normal wings. We had some pink eyes with little wings, vestigial wings. And we had some pink eyes with normal wings. And we got some numbers as well. So red eyes with vestigial wings, we had 125. Red eyes with normal wings, we had 376. Pink eyes with vestigial wings, we had 41. And pink eyes with normal wings, we had 117. And it's asking us which characteristics are dominant and which characteristics are recessive. Good rule of thumb, whatever is demonstrated in the F1 generation will be your dominant alleles. So here we can say that the dominant trait will be red eye and normal wing. It's only when we intercross these heterozygous flies that we get the range of different phenotypes and genotypes. Another little bit of information that, even though it's available and it's obvious by the F1 generation, if you look at the F2 generation, what is more expressed are the red eyes and normal wings. So it's a good indicator that that's the dominant alleles as well. Then asks us to come up with some symbols for use in our Punnett squares. And the best thing to do is to find your dominant alleles and choose those letters. So we'll have R for red eye, little r for pink eye, okay. And we'll have N for normal wing and little n for vestigial. These are good choices because it's very obvious to see which are the dominant alleles and which are the recessive alleles, because there's a big difference between those letters. It then asks us to draw a suitable genetic diagram to explain the results of the experiment. Now remember, we're talking about dihybrid inheritance here, so we're not just looking at one allele pair, we're looking at two allele pairs. But it's the exact same thing, it's the exact same principle. The first thing we've got to do is decide what parental phenotypes are going to be. Now in this case, so the results of this experiment, we're looking at the F2 generation, so we're going to be looking at red eyes with vestigial wings and pink eyes with normal wings. So if we've got red eye, vestigial, that's our pure breeding parent, and pink eye, with normal. So we've got the red eye, vestigial wings, pink eye, normal wings. These are our parent flies, okay? These guys up here. Oh, guys and girl, I suppose. What are the gametes going to be? Well, when we've got a pure breeding couple, there's only one option that we can have. It's going to have one dominant and one recessive. Same thing here. It's going to have one recessive and one dominant gamete. Why are they circled together? Because both these alleles are going to be in the haploid gamete. So that's one gamete and that's another. Okay, we're not going to put two circles around them because these two alleles will be found in the same gamete. What will the offspring of these two look like? Big R, little r, big N, little n. That will be our red-eyed, normal-winged F1 generation. Alright, red-eyed, normal-winged F1 generation. Now the next thing that they do though is that they get two of these and interbreed them, which means we're going to have slightly different gametes. We're going to have our red-eyed, normal-winged male crossing with a red-eyed, normal-winged female. We need to figure out what the possible genotypes are for the gametes. You could have homozygous dominant. You could have heterozygous recessive for wings. You could have heterozygous recessive for eyes, or you could have homozygous recessive. So these are our four possible gametes. Now, because we're crossing a male and a female with the same genotype, we can just assume that they're going to be the both. So these are my F1 gametes. How do we find the F2 offspring? How are we going to find the probability of the F2 offspring? For that, we're going to need a mega Punnett square. And I feel like Neil Buchanan having pulled that out, but still. What we're going to do is we're going to make our Punnett square with the alleles from the F1 generation, which means we're going to have homozygous dominant gametes, heterozygous dominant for eyes, heterozygous dominant for wings, homozygous recessive. And we'll do the same thing down the side. Homozygous dominant, heterozygous dominant, heterozygous dominant, homozygous recessive. Now for the dihybrid crosses, we now need to figure out what the potential offspring are going to be for each of these, but remember these are one gamete, so we're going to be having 4 symbols in each of these squares. By convention, we put the two alleles together, so it won't be big R, big N, big R, big N, it'll be big R, big R, big N, big N. Big R, big R, big N, little n. Big R, little r, big N, big N. Big R, little r, big N, little n. And we do the same thing. It's a bit like knitting, it gets therapeutic after a while. Again, I'm just crossing my alleles, doing the R's first, and then the N's. You can see that when we look at just 2 genes, it becomes a lot more complicated. Okay. Now we've done our crosses. Now we can see the probabilities of each of the genotypes of each of our F2 generation. The next thing they ask us to do is to work out the, so I'm just finding the question, so diagrams to explain the results of this experiment. So what we now need to do is look at the probability of each of these happening and match those up to the actual questions, the actual results that were given from the experiment. So we need to look at the phenotypes of each of these possible matches. Here we're going to have red and normal. Actually, do you know what I'm going to do? Just for a second, I'm going to make sure that I've got my codes here for reference, so you can see where I'm coming from. We've got dominant R here, so it's going to be red, and we've got a dominant N here, so it's going to be normal wings. It's going to be red and normal wings, red eye, normal wing, red eye, normal wing. Again, red eye, normal wing. Here we're going to have red eye, vestigial wing, red eye, normal wing, red eye, vestigial wing, red eye, normal wing. You can see the trend here. Pink eye, normal wing, pink eye, normal wing, red eye, normal wing, red eye, vestigial wing, pink eye, normal wing, and pink eye, vestigial wing. After we've done that, then all we have to do is count up the amount of different genotypes, different phenotypes. So first off, I'm going to count red eye and normal wings. So I have a little table over here in the corner. Sorry, it's getting a bit cramped, I know. Red eye, normal wing, 1, 2, 3, 4, 5, 6, 7, 8, 9. Okay, so I've got 9 red eye normal wings. I shall do red eye and then vestigial wings next. 1, 2, 3. Next possibility, we'll do a pink eye with normal wing. Pink eye with normal, pink eye with normal, pink eye with normal. So that's 3. And then we'll do the double homozygous recessive, or is there yeah, no, that should be it. There's only going to be one pink eye with vestigial wing down here in the corner. Okay, so red eye, normal wing, we had 9. Red eye, vestigial wing, here we had 3. Pink eye with normal wing, we had 3. And vestigial, we had 1. So we could interpret the probabilities that we've been given here, the probabilities we've been given here, and the actual data we've been given here, and we can see that they're roughly, there's sort of an indication that might be the right proportion. Now, how is it that we can tell whether or not this is accurate, that our probabilities are accurate? How likely is it this is just down to random chance? Because remember, these are just intimations, these are just indications of probability. This does not guarantee that if these two parents had this many offspring, that those offspring would have exactly those proportions of characteristics. This is just a probability game. If you increase your sample size, then you're going to go closer and closer to what the theory says is true. In this case, we only had a couple of hundred offspring, so perhaps the ratios aren't going to be 100% accurate. But that doesn't matter. We haven't been told to talk about that. We can have a look at the statistical analysis that we'd use for that later on. But for right now, it's just talking about drawing 2 suitable genetic diagrams to explain the results of the experiment, and we have done that. These genetic diagrams will explain the results of this experiment. Sorry, that's the postie.